Now I'm very pleased to introduce Thea Klansik today. Thea is a well-traveled scholar. In 2008, she left her home country, which is Slovenia, and started her bachelor degree abroad at the Edinburgh Napier University in Scotland. In her final year research project, Thea studied the impact of exercise intensity of salivary antimicrobial proteins and received a bachelor degree in exercise physiology in 2012. After that, she went to Technical University Munich to pursue a master degree in nutrition and biomedicine. She defended her master thesis in 2014 after spending 2 years looking into the impact of high-fat, low-carbohydrate diets on plasma lipid profile and inflammatory status of distal ileum of rats. 2015 was the year Thea decided to join Dr. Raylene Reimer group to investigate the obesity risk and early life antibiotic or prebiotic exposure here at the University of Calgary. Now, her current research interest is on the prevention of obesity and comorbidities. This involves programming in early life, during which the susceptibility to perturbations is high. In the future, Thea would like to work in industry and focus on improving the existing pharmaceutical products on the market. Specifically, she wishes to redesign formulas for children to decrease the risk of diseases such as obesity, allergies, asthma later in life. For those who know Thea, she is an animal lover to the point that she follows every dog profile on Instagram. She also enjoys night photography. Not even a cold night of minus 25 degrees Celsius can deter her from going out for a good photography session. Now today, Thea is excited about sharing the results of a first longitudinal PhD study entitled Early Life Antibiotics and Prebiotics, Impact on Gut Microbiota and Obesity Risk. Please help me to welcome Thea.
Thank you, Anquan, for such kind introduction. And I would like to thank everybody for coming. I feel very honored to be able to present here today. So as Anquan said, my first PhD study focused on indirect antibiotic exposure, meaning we gave antibiotics to pregnant and lactating dams, and then we monitored the obesity risk of their offspring. So I'll start with the introduction. So obesity is a worldwide problem. Currently, there's 500 million obese adults and 42 million obese children under the age of 5. From the 80s, obesity rates more than doubled. So if we look at the map of the United States back in the 90s, you can see that the map is mostly blue, indicating obesity rates from 10 to 14%. If we fast forward 20 years, in 2010, there is no such state anymore with such low obesity rates. Every country, every state in the country has obesity rates higher than 20%. In some extreme situations, here down southeast, obesity rates are higher than 30%. So the question we have to ask, what's happening? Why is there such obesity epidemic all over the world? And I can explain it with this graph, which is simplified version of the complexity of obesity. So essentially, there are several reasons why someone becomes obese. There's so-called physiologic adaptation. So during pregnancy, it's okay to be obese or overweight in the 3rd trimester of pregnancy. There's also so-called gene-induced obesity. That means that our genetic background will increase or decrease our risk of obesity. One extreme example would be leptin gene. Leptin gene, if the child is deficient for the production of leptin, they become morbidly obese. And the only way to help them is with leptin injections. Now, there's other hundreds and hundreds of genes which can increase your risk of obesity, even though most of them, the increase in risk is slight. However, if we have a lot of genes, the effects add up. And that might distinguish my risk from someone else. Third is diet-induced obesity. I mean, that doesn't need any introduction. I think it's very common that we now are exposed to a Western diet, high fat, high sugar diet, energy dense diet. So if we have a very high caloric intake, it will lead to increase in adipose tissue. Now, what I want to focus on today is so-called microbe-induced obesity. That means that microbes residing in our gut are responsible for us to be gaining more weight than normal. And why does this happen? Our gut is very sensitive early in life. So we can have, let's say, a C-section or antibiotic exposure, which is part of my study, which leads to perturbations in our gut and can lead to increased energy harvest, meaning if a person which has microbial dysbiosis and a person which has a healthy gut and healthy bacteria present in their gut, if they eat the same meal, the other one will be gaining more weight, simply because their bacteria are able to extract more energy from the same meal. Another example how microbes can increase our fat mass is through metabolic changes in metabolic signaling. The main organ here I want to mention is liver. So liver is the main metabolic organ, and it can lead to changes in hormone productions. It can lead to increased adiposity in liver, and if there is increased adiposity in liver, liver will lose the sensitivity to hormones, glucose, etc. Last but not least, when it comes to microbial disturbances, is changed inflammation. So if we have changed immune function, we enable to store more fat. So for example, when a person is obese, they are also most likely all the time under this low-grade inflammation. And that happens because our gut loses the ability to be tight. So bacteria in our gut should not pass into our bloodstream. And if they do, the liver is the first organ which should take care of it. But if the liver is inflamed or if there is a lot of fat present, that bacterial species such as LPS, which is lipopolysaccharide, it's a part of bacteria, can go to a systemic circulation and leading to overactivation of our immune system, low-grade inflammation, and excessive fat storage. So as you can see, it's all interconnected. So what is gut microbiota? Most of the research focuses on bacteria, but now also fungi research is emerging. So gut microbiota in our gut consists of viruses, fungi, protozoa, and bacteria. More than 90% are bacteria, so I guess this is why most of the research focuses on it. But it's not just gut microbiota. We have bacteria pretty much everywhere. There's bacteria on our skin, there's bacteria in our mouth, there's bacteria in our eyes, so they're covering our surfaces. However, the most densely colonized ecosystem of the human body is in our gut. And they give us functions which we as humans normally don't have. So if we talk about genes, bacteria in our gut have 100 times more genes that we have. So they have ability to provide us with metabolic functions that we as humans don't have. One of them would be vitamin production. So why do we study early life? Think of your gut bacteria as the organ which develops after you're born. So the first 2 years of life are critical. Why? Because early in life, the diversity, meaning the number of different bacterial species in our gut, should be low. And that gives us benefits for the health of a child. So we should keep low number of different bacteria. However, that also means that the stability, the bacterial composition in our gut is very unstable. So if we hit them in the first 2 years of life in humans with antibiotics, or if the baby is delivered through a C-section, their starting point and development will be disrupted. And that can have severe consequences later in life. We study obesity, but we do also know that if you disrupt microbial composition in the first 2 years of life, you predispose a child to asthma, allergies, type 1 diabetes, just to mention a few. So antibiotics are the top prescribed drugs in children. So if you look at the list, the top 6 drugs prescribed to children from the age of 0 to 17 years, 5 of them are antibiotics. The only one not antibiotic is albuterol, which is an asthma medication. And when we look on the average statistics in the United States, by the age of 2 years, on average, a child in the United States has been given 3 courses of antibiotics. By the age they are 10 years old, there have been given 10 different courses of antibiotics. By the time they're 20, 17 courses of antibiotics. So it's very clear that we need a system of giving something else to reduce the risk, which is like a side effect of giving them antibiotics. Once we combine the two, if you remember this map, this is obesity rates. And like I said, this is data from 2010. If you look down southeast, obesity rates are higher than 30%. When we look at the prescription rates of antibiotics in the United States in 2010, the picture is very similar. The darker the color, the higher the prescription rates. So the highest prescription rates kind of overlap with the highest obesity rates. And I know this only means association. However, the group of researchers from New York did very cool experiments in mice when they took a mouse which was conventionally raised, so they had their own microbiota. They gave them low doses of penicillin, and they took that fecal matter and transplanted that fecal matter to a germ-free mouse. Germ-free mouse simply means that that animal is sterile. So they took the feces from the antibiotic-treated animals, they gave it to the germ-free mouse, and that mouse became obese. So it's very clear, at least in animal studies, to say that antibiotics can cause obesity when given to animals. So the question we asked in our lab was, could we prevent obesity associated with early life antibiotic exposure? And working in Reimer lab, we decided to go with prebiotics. So what are prebiotics? Prebiotics are non-digestible food ingredients which selectively promote the growth of health-promoting bacteria in your gut. So if we look at the equation, we cannot digest prebiotics as a human because we lack enzyme to digest it. However, bacteria in our gut possess that enzyme, and once prebiotics interfere with bacteria, fermentation occurs, and there's short-chain fatty acid production. And that is the actual health benefit of bacteria. Short-chain fatty acids would be butyrate, which feeds your epithelial cells in your gut, and it's very important to keep your gut healthy and not leaky for the bacteria. The second example would be propionate, which targets the liver. The third one would be acetate, which targets the brain. And there is, of course, some side effects, such as bloating and flatulence, and that is because there's gas production during the fermentation process. So what are the health benefits? In human and rodent studies, we were able to show that prebiotics reduce body weight, reduce fat mass, reduce inflammation, improve metabolism, and increase satiety hormone production. Essentially, everything we want to fix after antibiotic exposure. So the question got slightly longer. We simply added prebiotics to the equation. So can we prevent obesity associated with early life antibiotic exposure by co-administering fiber in their diet? This is the way we designed the study. So we got the rats, we mated them, and in the 3rd week of pregnancy, which is the 3rd trimester in rats, because rats are only pregnant for 3 weeks, we started giving them either antibiotics. Second group got antibiotics and a prebiotic diet. The 3rd group got prebiotics. And the 4th one was just control on water and a normal diet. And we kept them on intervention until the end of lactation, which lactation period in rats is 3 weeks. So altogether was 4-week intervention. We then sacrificed the dams. We took their offspring forward. We put the offspring on a controlled diet to assess their early life growth rates. And as they reached 10 weeks of age, we put them on a high-fat, high-sugar diet, and that served as a metabolic challenge. So essentially, we had to challenge those animals to see, are they really more prone to obesity? I hope you, I just want to point out at this point that the only animals exposed to antibiotics or prebiotics were dams, so mothers. The offspring was never directly exposed. So what were results in dams? This was a nice surprise for us because this was never published before. Normally, when studies like this are done, they talk about the offspring. They never described what happened to the dams, what happened to the mothers. But we routinely recorded body weights, and we, of course, collected tissues, and we were nicely surprised with the results. So this is the body weight, and this is gestation period, and this is lactation period. So the intervention started gestation day 15. We gave them antibiotics until the end of pregnancy, and then as soon as they gave birth, antibiotic dams, as indicated in the blue, didn't lose as much weight as they should. And they remained significantly heavier throughout lactation. Whereas as soon as we gave them a prebiotic diet with their antibiotic, focus on the green, you can see that the body weights after giving birth were completely normal and comparable with other groups. At the end of the intervention, here at weaning, we did DEXA on those animals, and we looked at their body composition, and we saw that dams given antibiotics had higher body fat percentage. However, it was completely reversed with the prebiotic diet. As said before, with prebiotics, you should reduce your food intake. Prebiotics are very strong inducers of satiety hormones. So it's logical that in gestation week 3, when we gave them prebiotic diet, we could see that rats given prebiotic diet were eating less per day. And that might partially explain why those animals were so much lighter than our antibiotic dams. But what doesn't explain is, with the food intake, is why were control animals eating the same amount as our antibiotic animals, but the weight difference was still there. When we looked at the hormones in those animals, blood was collected from the portal vein at sacrifice after 12-hour fast. We could see that whoever was given antibiotics had high leptin, low GLP-1, and high insulin. And that is very common if you have some metabolic impairment. So those animals were metabolically impaired. And could we fix it with a prebiotic diet? We were able to reverse leptin, which is great news. We were able to increase GLP-1, which fits nicely with the data of food intakes. High GLP-1, low food intake makes sense because it's a satiety hormone. And then insulin, unfortunately, we weren't able to fix with a prebiotic diet. Once we did functional tests assessing the glucose tolerance, we realized that prebiotics were protective and they improved glucose sensitivity. So 30 minutes after the glucose load, you can see that animals on a prebiotic diet had lower glucose levels, meaning they were able to deal with glucose load better than the other animals. And one of the hypotheses was, well, maybe their liver is healthier, maybe they have less fat in the liver. So we looked at the liver triglyceride levels, and we could see that prebiotic animals, whoever was given a prebiotic diet, had lower liver triglyceride levels, which nicely fits with the data from glucose tolerance tests. Now we come to the bacterium. So we collected feces longitudinally. So we collected at gestation day 8, which is before the intervention happened. We collected it at birth, which makes sense because this is the actual crucial time point, because if our gut microbiota develops at birth, the starting point is extremely important. And then we collected it meet the intervention, and at the end of intervention, we did different kind of analysis, and I'll present results after this slide. What I want to show you here, though, is at birth, Lactobacillus, which is the key bacterial species in the vaginal canal, was absent in antibiotic group, meaning that the key species were not present at birth. It could mean that whoever was given antibiotics didn't have the right starting point of development at birth. When I did some research, and I was like, well, why did this happen? I couldn't explain it. But then I realized that low doses of penicillin actually targets mostly gram-positive bacteria, and lactobacillus are gram-positive. Now, if you target gram-positive, what's going to thrive? Gram-negative, I guess, right? So at lactation day 1, we could see like a spike of this Enterobacteriaceae in the antibiotic group only. So again, it shows how microbiota was imbalanced in antibiotic groups only, and it was treatable or preventable with the prebiotic diet. If you look at the green line, there is no differences with other groups. Now, prebiotic diet promotes the growth of health-promoting bacteria, and the one key species is Bifidobacterium. And Bifidobacterium, you can see the longer the animals were on the diet, the higher the Bifidobacterium levels were in those animals. So that was just the qPCR data. This is the fancy stuff. So this is the 16S rRNA sequencing. So essentially, we got the samples at sacrifice, we collected fecal matter, and we extracted DNA and sent it away for sequencing. And the difference between qPCR and sequencing is that with sequencing, they sequence everything. So with qPCR, I decided what I want to see, but here, it's actually the overall bigger picture. So on a phyla level, it's like looking at the universe. We have big groups of bacteria consisting of a lot of different types. And if we group these bacteria on the phyla level, you can see that prebiotic groups have this green part, which is not present in the other groups. And again, it was pretty exciting to see that this fancy results matched our qPCR data. So actinobacteria were significantly higher in prebiotic groups, and within that group, we have bifido. So when we actually looked at the numbers, what was the relative abundance in those groups, we could see the control and antibiotic group had a relative abundance lower than 1%, whereas prebiotic animals had abundance of 20% and 33% respectively. So the difference was really, really high. Okay, so that are the mothers, and now we're going to focus on the offspring. Like said before, the offspring was never directly exposed to antibiotics nor prebiotics, only their dams. So we took them at weaning, and we put them on a control diet to assess the growth rates, and then we challenged them with a high-fat, high-sugar diet to see whether they're really at higher risk of obesity. Already, immediately after we separated them from the mothers, we could see that males and females from antibiotic dams, you can see the blue lines, just look at the blue lines, they were growing significantly faster than any other group. So those animals had increased growth rate, even though their food intake, in terms of daily food intake in kilocalories per day consumed, was identical between the groups. So the amount of food they ate cannot explain why antibiotic offspring was growing faster than the rest of the groups. And what was really exciting was to see this, because we do know that if early life growth rates are increased, those animals are very likely to be obese later in life. It's a good indicator of the obesity risk. Once we actually challenged them with the high-fat diet, so they were on a control diet for 10 weeks. At 10 weeks, we started a high-fat, high-sugar diet, and we kept them on a diet for another 8 weeks. 5 weeks was sufficient to see differences in phenotypes. So antibiotic males were gaining significantly more weight already at week 5 on the intervention, and it just got progressively worse. And we performed DEXA at the end. So week 18, we performed DEXA on those animals, and we can see that the reason why those animals were heavier was because of the increased fat mass. Just what we see in the mothers. And it was preventable. Again, prebiotic diet of the mothers was able to protect the babies as well, was able to protect the offspring because they had significantly lower fat mass than antibiotic group only. What happened to the female offspring? The picture is very similar. So you can see that we did the same thing when they were 10 weeks old. We started challenging them with a high-fat, high-sugar diet. Immediately after 5 weeks on the diet, they were gaining significantly more weight than other groups. However, when we performed DEXA, there was no significant difference between the groups. However, it's very clear that there was a trend. And because females are smaller, the difference in weight was only about 30 grams, and 20 out of 30 was due to fat. So I still think it's relevant. It just didn't reach significance because they're just growing slower than the males. What happened in terms of metabolism? It's very cool to see that there are sex-specific differences. So before the high-fat diet metabolic challenge, male offspring coming from antibiotic dams was already insulin resistant. So insulin tolerance test is essentially you inject the animal with insulin and then you monitor their glucose levels. And the lower the glucose levels, the more sensitive they are to insulin. So you can see that the blue line on the top indicates that there was insulin resistance in the male offspring. Now with the females, there was no difference at week 10, but after they've been given a high-fat diet, they developed insulin resistance. And you can see that antibiotic females were insulin resistant compared to other groups. And again, like with dams, it was preventable with prebiotic diet. Last but not least, this is the last slide I have in terms of results. If you remember, the dams had a really high levels of those bifidobacteria, which are so protective for the health. And you can see that at weaning, as soon as we separated the offspring from the mother, we could see higher levels in prebiotic groups also in the offspring. And the differences were gone as they were exposed to the diet. So in summary, if we give antibiotics to pregnant lactating rat, they don't lose as much weight as they should when giving birth, and they remain significantly heavier throughout lactation. There are microbial disturbances and metabolic ones. And what's worse is that their offspring is very likely to be obese later in life. However, if we give them antibiotics and a prebiotic diet, we induce the normal weight loss after giving birth, and we also protect their offspring, which remains leaner after challenged with the high-fat, high-sugar diet. I would like to thank my amazing supervisor. Dr. Reimer. Oh, God, I'm so sorry. Yeah. Thank you. And I would like to thank my lab mates, who are also my friends and funding sources. Thank you very much. Sorry. Thank you.
What kind of antibiotic did you use, and is there a difference if you switch them?
Great question. I used low doses of penicillin. The reason why we used penicillin is because it's very commonly used during pregnancy, in humans as well. And the second reason we used it is because previous studies in mice have shown antibiotic is the most likely to give obese phenotype. That's the reason.
I guess I have two questions. The last one. How often do pregnant women get prescribed antibiotics? Is it at the same rate as the general population?
It's very high, actually. There is so-called strep B infection, even if there's just an assumption someone is strep B positive, they will be given antibiotics, even though it's only true in 2% of cases. So to answer your question, 50% of pregnant women get antibiotics a day before delivery, and they're flushed or during the delivery because of the suspicion of this infection. But there are also antibiotics. There's more women who get antibiotics in the second and third trimester of pregnancy. So it's very commonly prescribed.
So if you have a C-section, and then that's related to they don't develop this microbe, they don't get the bacteria from going through the birth canal, why can't you just take fluid from the birth canal and smear it all?
That is a fantastic, actually it's happening. It's called smearing. So, no, it's true. Dr. Knight from the United States is a big researcher in microbiota field, and his wife was about to deliver, and they were really against the C-section, but of course there was an emergency C-section, and he put a gauze into his wife's vagina, and then he smeared the baby, and up to 3 years of age, the baby had no infections, which are very common in children. Now, this is an N of 1, so means nothing, but at the same time, there are a lot of clinical trials happening right now where the women who know that they're going to deliver through a C-section actually smear the baby with the gauze. Of course, they have to screen it and make sure that doesn't have any viral infection or bacterial infection. But if that goes through, you can smear the baby and you hope for that the infection will be prevented.
Yeah, I was
Hey.
just wondering about the length of the antibiotic protocol. So I feel like 4 weeks seems like longer than a doctor prescribed in the person. So what was the basis for that?
Right, so that is a great question. The reason why we went with a 4-week protocol is because we administered FDA-approved dose, which is 100 times lower dose that you would give to a woman. So and again, I'll refer back to Dr. Blazer's study. That type of administration seemed to mimic microbiota profiles of human data, and it seemed to mimic the phenotypes, so increased fat mass, increased body mass in the best possible way. So for that reason, we administered it for 4 weeks, and I agree that that's not a good translation potential in terms of human. So for that reason, for our second study, we are giving direct antibiotic exposure of azithromycin, and we're giving it in 3 human type doses and human doses. So it's a higher dose, it's a short exposure, and it's only 3-day protocol 3 times. So that mimics human administration completely.
You said that antibiotics like decrease the diversity in gut microbiota, specifically like at the younger age. What promotes diversity?
I don't know. Did I say I don't think they decrease diversity.
Sorry, I should phrase it right. So you said the diversity is highest at those first crucial.
Diversity is lowest. Diversity is lowest when you're born. So, which is really funny because as we age, as we are adults, right, we should have a really high diversity to be protected against infections. Now, when you're a baby, you should have a very low diversity to be protected. Because if a baby, for example, it's not just C-section, if a baby is breastfed, their diversity is very low. Whereas if you give them formula, they have a very high diversity. If you perform a C-section, they have a high diversity. And at that point, scientists realize that high diversity in the baby is bad. So antibiotics, what they do is, like I showed the data for Lactobacillus, right? They might eliminate some of the key species in that critical period of time where we should have a low diversity because everything is so unstable. Yeah.
Thanks for your talk. Assuming that they got a 10-gram meal, what proportion of that 10-gram meal would be associated with prebiotic?
1 gram. So it's 10%. Welcome.
2 questions.
Sure.
One is, what percent of the antibiotic might end up in the milk or being transferred to the fetus by transport?
Great question. In this particular study, nothing. Why I'm so confident about it is because when you give penicillin to a pregnant or mother, to a human, the dose you detect in a breast milk is 100 times lower than whatever she took, 100 to 1,000 times lower. So there is an amount coming to the breast milk, but it's really low. But what we used was 100 times lower dose than prescribed to women. So in that sense, there was nothing most likely detectable in the milk.
And my second question relates to the milk itself. Supposedly it has a lot of antibodies to bacteria. There's a migration of lymphocytes from the gut to the breast and actually contributes specific antibodies. So how does the breast milk actually influence the colonization and the stabilization?
Oh, it's the best thing you can do for the baby. So if a mother can breastfeed, they should, because it's a crucial component. Whatever you'll be feeding in the gut, that will grow, right? So like you said, baby lacks immune system and it gets it through the mother. And bacteria, colonizers as well, they're giving through the maternal milk as well to the baby. So I think the role is extremely, extremely high for the development in the first few years of life. So it's very important.
When the offsprings born, you couldn't explain why they grew fast, yeah?
Yeah.
Because they received the same amount of food. What about, I don't know if you check the growth hormone? You didn't.
No, I didn't.
This can explain why the antibiotic would grow faster than the prebiotic.
It could. It also could be like activity level, right? I didn't record activity levels. It could be, like you said, a growth hormone. Unfortunately, we didn't bleed the animals at 3 weeks of age. So I can't even check, even if I would want to. What would be maybe interesting to see is do a bit of more sequencing and look at microbiota composition, because it also could be that their microbiota composition was changed in a way that the extraction of calories from the same amount of food was different. So I'm hoping to look more at that and also look at fungal analysis, because research has shown that if you eliminate certain type of bacteria, and in my case, Enterobacteriaceae were thriving at birth, that bacteria also enable fungi to grow. So that would be also one thing to look at. Yeah, Walter.
So has ever anybody looked at children born by C-section compared to children born normally?
Yeah.
And the obesity rate is higher with C-sections?
Absolutely. And right now, like it was shown in humans, in animals. And what's interesting, when it comes to antibiotic exposures, the boys are the ones which are more affected. So the boys will be growing faster, more fat accumulate, and then the girls are slightly less. Whereas when it comes to C-section, the females, the girls, are more likely to be even more obese. And now there's just a paper which came out in Science Advanced, and they did a very cool study with C-section only, because the problem with human studies is if you have a C-section, you'll get antibiotics too. So how can you separate the two? What's causing what, right? So in that study, they delivered mice through a C-section, they cross-fostered them, and whoever was delivered through a C-section had increased growth rates and was gaining the weight difference was even more striking than with our stuff.
I read once about C-section rates, and it seems that the Netherlands is the lowest, is, I don't know, 3, 4, 5%, and Brazil was the highest, is way over 50%.
Rio de Janeiro over 90%,
Yeah.
90.
So is that, so that then would presumably have an effect on entire countries then?
Absolutely. So I think, like, I spoke to my friends from Brazil, and I was like, what? Because there was a talk in Banff, and they said C-section rates in Rio de Janeiro are the highest, I think, in the world, over 90%. I'm not sure if she said even over 95%. So essentially, the only one who doesn't deliver through a C-section is the one who gives birth in a car on the way to a hospital. It's very problematic, because more and more research is coming out. It's not just obesity, it's asthma, it's type 1 diabetes, it's a lot of other stuff, and those diseases are on the rise. And we are also increasing the antibiotic consumption, C-section rates, so we should really rethink this process. But I guess doctors like to schedule a C-section is way easier than a natural birth. That was the answer I got from friends from Brazil. So
if you give fecal matter of one mouse to these
germ-free mouse, yeah.
how do you do that?
So those germ-free animals are delivered through a C-section.
I know how germ-free
Okay.
animals are. How do you inject it?
No. You gavage. You prepare the cocktail. It's kind of yeah. You do glucose tests with a gavage in the stomach. That's it. They do it in humans as well for the C. diff infection. The success rate to treat a C. diff infection is over 90% with a donor of a fecal matter. So the lineup in the United States is really long, because they need to screen you for everything. But if you have a C. diff infection, you're bound to the toilet for the rest of your life, or you get a fecal transplant, because the success rate of antibiotics is only 30% to treat it. So 70% is likely that you will still have issues.
That's why I come to the seminar. Fecal transplants. Why are the female rats so much leaner than the male rats?
Hormones, maybe? I don't know.
In humans, it's, I guess, accepted that it's the other way around.
Unfortunately, I think in humans is because of the fact that we have to carry children. So it's safer for you to survive, to have more fat mass. Whereas in animals, I don't know. I do know that why my data with females is slightly more messy, and I guess that's why people like working with male animals more, because there's no menstrual cycle. I didn't control 200 rats when their periods is gonna come, because it would be too much work and I don't even know how to do it in rats. But I only controlled on the time that they were born, but of course they don't mature at the same exact point. So menstrual cycles will mean fluctuation of hormones, will mean like it comes with a lot of different things. So I guess why female animals are leaner, I just don't know, I honestly.
So it's my understanding that rats have one of the best, or maybe not even, or maybe even the best immune system for any animal. They don't really seem to get any infections ever naturally, they just are able to fight that. Whereas humans, of course, are not. So do you think your antibiotic intervention would have a very different effect on such an immune-resistant type of animal compared to humans?
It could be. I honestly didn't look at any immune parameters yet, but that's a legit point. It could mean that those animals have a better immune system to start with, and then antibiotics do not disturb them so much. I honestly don't know.
Were they already different at birth, or are they about the same weight at birth?
Same weight at birth, but at weaning, so first 3 weeks of life, control and antibiotic animals were identical. So there was no differences in, whereas prebiotic offspring was lighter already throughout lactation. And it could be that this prebiotic diet is also coming through milk and feeding the baby. So that might be one of the reasons why these animals were leaner to start with. But antibiotic and controls were absolutely identical. For that reason, we needed a high-fat diet challenge to see whether risk is really higher.
So you always say that, or you hear that in animals that we eat, like chicken, beef, whatever, there's always a lot of antibiotics. Do you think that that might impact this solution as well?
Yeah, I guess then, you know, my study of 4-week low doses of penicillin exposure makes perfect sense. I think that's a legit point, because there are different types of antibiotics, like you pointed out before. So penicillin acts like more locally, whereas you have azithromycin, which targets the tissues. So azithromycin, if they're giving that to the cattle, or if they're giving that to pigs, I'm pretty sure I get some dose in a steak later on, because the concentration in a tissue would be 100 times higher than in plasma. So, you know, whether that is still active, or how my bacteria react to it, if it's no longer viable, I cannot go there. Like, I don't know, but it might have something to do with the fact that there is a lot of obesity worldwide.
In the sterile rats that got the fecal transplant, did you say they became more obese?
Yes, so they did experiment, and what's really cool about that experiment was a proof of concept, because they had a rat which was not given, they had a mouse which was not given antibiotic, they had a mouse which was given low doses of penicillin, just what I used, and then they put both of these fecal matters to a germ-free mouse. So before these guys even got obese, they were normal weight, they just got a dose of antibiotics, and then they took this fecal matter, put it in a germ-free mouse, and that mouse who was given microbiota with antibiotics was growing unproportionately faster than the other one.
And what was the mechanism behind that? Were they consuming more calories, like that? Were they less active, or what was the underlying
in terms of the microbiota like the only mechanism I can think of is different bacteria present in their gut, because they did measure their locomotor activity. They did measure the amount of food. There was no difference. And then of course they sequenced their fecal matter, and that was different because they got the microbiota treated with antibiotics, right? But they even did bomb calorimetry, where you can see how much calories was extracted from the food, and they didn't see any difference. So at that point, I was like, well, I don't understand.
on that, evolutionary speaking, wouldn't it be great that if we extract a lot of the calories from the food that we have? So
It's higher, increases your chances of survival, for sure. But that was only relevant back in the days when there was like no food available, right? And it's kind of sinking us now, I think.
but I mean that, yeah, but I mean that was the case for millions of years, and now for about 50 years we have enough food available at any given time.
And there's like antimicrobials are everywhere. Then we have like also what could change our gut composition, microbial composition, is also processed foods. You know, there's a lot of different factors coming together. And as I said before, it's very complex when it comes to obesity. I think it's a combination of a lot of factors. Yeah. And what's really interesting is when I was studying for my candidacy, I learned that in the 3rd trimester of pregnancy, your gut bacteria actually naturally change in a way that they are more able to extract more energy for the baby. So if I gave them antibiotics in the 3rd trimester of pregnancy, I could just delay the process of normalizing microbiota after birth. So that could be one of the explanations why my animals remain significantly heavier, because I just prevented the normalization of the gut after they've given birth.
So your offspring animals are in the same cage as the mothers for the first 3 weeks of their lives. Do the mothers still get prebiotic diets during that time? So how do you make sure that the babies don't eat any of that?
They, of course, do eat. They eat, I think, the last 5 days before I separate them, because there was no way of separating the two. Once they grow, they climb on the food cup, but I did double. I put another food cup under. I taped it with a duct tape so they couldn't reach it as long as possible. But the last 2 days before I weaned them, clearly there were occasions when I found a pup in the food cup, and I was like, you gotta be kidding me. But yeah, they could be consuming their diet a little bit as well. Hopefully no drinking though. I don't think they can reach the bottle.
Scott, could you show us the slide with the breakdown of the RNA sequence?
Yeah. If, I don't know. It's RNA-seq, this one?
Yeah. So at the phylum level, it seems like the control animals are dominated by this red phylum.
Firmicutes.
The antibiotics and the prebiotic group. You have this green, you were pointing that out. So I assume this green phylum or phyla has to displace other bacteria, right?
Oh, absolutely.
So if you look, phylum is like right below kingdom, right?
Yeah, yeah.
So if you started looking at higher, like what are they displacing?
So.
Because it's still very different than the control.
Oh, absolutely.
Before you answer that,
Yeah.
can I actually ask, are they actually displaced? Because you give relative values. It could very easily be that the rate in the top bar, in absolute terms, might be bigger than the rate on the lower bar, depending on what the total amount is.
I think these competitive systems tend to be at their carrying capacity.
Yeah, I mean, I don't know that. I'm asking the question.
Sorry, I didn't understand. Could you repeat?
Well, he asks, you're displacing the rate on the top bar because you have less. And my question is, do you actually have less? Because if you have 3 times more of these bacteria, then you still would have more rate in the top compared to the bottom because you only give relative.
I understand what you're asking, but the answer is the concentration of the DNA we send for sequencing is normalized. So
you're comparing
exact amount of bacteria, and this is the composition relative to the total count. And Scott, to answer your question, of course, there's way more. To be fair, there's 12 pages of the Word file where the results are significant between groups, and I only picked one. I picked, and I'll explain why. I picked Bifidobacterium because the differences were the highest. Sometimes the results are still significant, but we are talking 1% relative abundance versus 2, which might be still relevant for your health, but for the simplicity of this talk, I only put this particular genus on the genus level, but I have it all. It's like family, then you have class, genus, and then you go down to the most high definition OTU level. But I need to learn how to do bioinformatics to present that in a better way than a table. That's it.
I think there just might be something interesting in how the distribution shifts as you go down.
Yeah, absolutely. Yeah, it's 12 pages of results, and I didn't know how to make it like a bigger picture to visualize the differences between groups.
So the quote-unquote obesity epidemic has been reported to be partly diet and partly sedentary behavior. So does exercise actually change the microbiota? Because, I mean, the animals are living in your hotel.
That's true. True. I'm not familiar with any particular study that would be looking at the exercise and then how does it change your microbiota composition. There was a few papers coming out that would describe the differences, but it's really hard to control in terms of diet, and when it comes to humans, you need really high numbers of participants. What makes this research even harder is I feel like when we look at the microbiota of people in this room, we are all different. So we all, like, our microbiota is different as our as our fingerprint. Like, it's very different. There's a core microbiome, meaning we have some similarities, but if we go down to the OTU levels, we all are extremely different. So that makes human research much harder. But exercise studies are definitely on the rise now when it comes to microbiota sequencing and like looking at bacterial composition and how does it change with exercise. I also feel that people who are exercising are generally eating healthier. So you feed them fiber, it's gonna change. I can't see you. That's okay.
Did you notice any difference in milk production between the different protocols? I feel like that could be something very relevant in humans, particularly like women having to feed their babies formula and then.
For I can give you human data. I don't know about my rats because I didn't measure how much they're lactating. But in humans, if you're obese, your milk production is reduced. And it seems like the milk is more concentrated. So it comes with a lot of issues. So there's a lot of leptin in obese mothers. Like, there's a lot of leptin in the milk. And what seems crazy to me is that leptin also programs your hypothalamus to form the projections in the brain of a baby. So again, if you skip the breastfeeding, you might be programming the baby the wrong way because they don't get the necessary signals to get the brain projections developed in the way they should. So yeah, in humans, obesity definitely impacts the milk production, but I don't know about the rats.
I have a question.
Yeah, sure.
So, what is the GLP that you showed?
It's an incretin. It's a hormone secreted from your gut after you eat, and it targets the brain in order for you to stop eating.
Oh, so the higher the value, the better?
Yeah.
Oh, so there's no like a value that you pass that it
No.
will be harmful to you because you don't eat anymore?
No, I mean, this...
Because in your, can you go back to the results? I have that questions because there's some...
GLP, there we go. Yeah.
So if you, I presume the control is a good one?
Control, yeah, it's control. I don't know if it's a good one. But I do know that in this case, in this case, prebiotic is a good one because they were leaner and they were eating less.
Oh, okay.
So, and the reason why they were eating less could be explained by the higher level of GLP-1 because they feel full faster. So these hormones, they act on your hypothalamus and they make you stop eating, essentially.
So if you are training to get bigger, you want more nutrients, protein. And if you have too much of this GLP, would it inhibit your growth muscle?
Oh, I honestly don't know. Like if GLP-1 has any impact on the growth? No. It's more appetite, right, Raylene?
And there's redundancy in the system, so there's always a big group of hormones working together to make you either hungry or full. So if you were getting to the point where you're getting too thin, other hormones kick in to take over in the system. So there's a lot of redundancy built in. So you have to be really messed up with these hormones to ever get to what you're talking about and inhibit your muscle building
Really?
With your body mass now, I know you presented, I think it's in absolute values of grams.
For mothers, yeah, but for the offspring was the weight gain relative to week 4. Sure. Okay, these are the change in body weight from
The body fat mass.
here.
So with this, it's in absolute values of grams. If you look at percent body fat,
There must be a reason I put the fat mass here, but I think there must be a difference, but probably it's not as big. So the reason why I put fat mass here is like if they gained, oh boy, if they gained like 40 grams more in the same period of time, my question would be, what was it? So if you look at this graph, you can see that the antibiotic animals had like 40 grams more fat in their body.
And the total difference is about 100 grams.
The total difference?
Is about 100 grams.
It's about 50.
50?
600 to 650.
From the lowest to the highest?
Oh, the lowest, that's a lean control. Yeah, that's about 100. And here it's like 100. So it works as well. That's nice. It's like, oh my God.
Was the lean mass the same?
Lean mass, I think there wasn't a big, lean control had more lean mass, but the rest of the animals were very similar, I think. I have no idea. I can show you later on my laptop, but I have the data, I just didn't put.
I have another question.
Okay.
Of course. So
Sweat.
I just recently read, saw that in the news. I mean, prebiotic, as you show, is great. You know, you control the obesity, and it sounds wonderful. But then I saw another study that looked into taking the feces from people with healthy microbiota, and then they make it into pills, and then ask those people who with obese disease to eat it. So it sounds gross compared to prebiotic, which sounds
Well,
which one do you think is better? Like, what is the difference between
Prebiotic and probiotic, okay.
and the one that's
So probiotic is a living microorganism. So essentially you would be eating bacteria, like encapsulated. I'm slightly skeptical about it because like if you eat encapsulated living microorganism, it has to survive the stomach, it has to get to your gut, and there it has to establish itself. So I feel like prebiotics are way easier for you to feed a bunch of health-promoting bacteria because we're talking about ecosystem. So if you're introducing a new animal to Banff, it might disrupt the whole forest and some animals would go extinct, and that's exactly what's happening in our gut. So that's why I feel like fecal transplants are so successful because you're introducing a group of bacteria which function together. So in order to maintain that fecal transplant, you need to feed them the right stuff. So in order to promote their health, I think the combination of both would be the best. So they even did studies with fecal transplants, and they looked in diabetic population. They did fecal transplants with so-called super donors. So yeah, they super healthy people, and they have ideal gut bacterial composition, and they were able to reverse insulin resistance for 6 weeks with one transplant. Now my question is, what if you would put these people on a prebiotic diet as well? So if you would be able to maintain that ecosystem longer like in your gut. Because if I switch the diet to a Western diet, I'm going to completely change my gut bacteria. If I switch to a prebiotic diet, I'm going to change it as well. However, as soon as I stop the diet, it's going to bounce back to whatever it was. So the gut is very resilient as we are older, but it's very sensitive when we are young. So
if you change your diet dramatically, how long will it take before you can change like a bad to a good microbiota, gut microbiota?
yeah, I honestly don't know. I know that already at 24 hours on the diet will change your gut bacteria. So if you go 24 hours on a fiber diet, high fiber diet, your bacterial composition will be different. But if you stop eating that, it's going to bounce back to whatever you were. So the key point is the only diet that works is a change in lifestyle.
So
you have to do it forever.
In other words, the gut microbiota that you have at a certain age essentially is stabilizer. So if you want to perturb it from that state, you have to change lifestyle. You cannot just do it for a little bit and then you establish a new one.
We wish, no, exactly. That's you should just maintain the diet, otherwise it's going to go back. And that's the resilience of the adult microbiota.
So, I mean, a lot of people get a colonoscopy where they get rid of all their microbiota, well, except for the biofilm parts.
Yeah.
Does it come back to what it was? Naturally? Because you still have bacteria there? Or it seems to me that that's the most drastic case I can think of.
That, or like if you're very sick and you're in a hospital and they flush you with all sorts of antibiotics, you kind of become germ-free, like not germ-free, but like your diarrhea would completely eliminate bacteria from your gut. And the problem with that is hopefully you can restore it back to what it was. But in some cases, the C. diff infection I mentioned before, when you're in a hospital, there are so-called opportunistic bacteria. When given the space, they will grow. And C. diff is one of those bacteria that can really mess up your gut in a way that if you are deficient in certain bacteria, because if they don't no longer occupy the space, then those opportunistic pathogens start growing, and it could be really dangerous if you're like in a hospital, and then you really need a lot of different treatments because they are antibiotic resistant. So it is an extreme situation. Like even those colonoscopies, like it's an extreme situation that it would go so wrong, whereas in children, even diarrhea can be lethal. Like if it's a newborn, if they have diarrhea for a few days, they might die because there's just no way that they could produce enough energy to grow. Whereas when we are older, it's easier to bounce back. I think that's enough. Thank you.
Now, before we end this seminar, I would like to invite Dr. Brent Edwards to make an announcement. It's an emergency one.
Great job. I just want to take a sec to acknowledge Walter Herzog, our fearless leader, who has made an incredible, has received an incredible award recently. He was one of 5 scientists in all of Canada to win the Killam Prize. This is, for those of you that don't know, this is the highest award that you can achieve in Canada. So it's as a scientist, I should say. So very well done. Last time anyone at this institution won the award was, I think, 1991. So again, great job, well done. You make us proud every day. And the Nobel is next. Thanks.
Very good. Next week, please come back, join me, because Dr. Richard Willey will visit from the University of Montana and will tell us about the model learning strategies to optimize gait restraining. I'll see you next week.